very much. Thanks to everyone that I'm here. I'm glad to be here. I'll just quickly, tell some of the, work that we are doing. Of course, in a career of more than 15 years, you do various things. Some funny things, some are not, but one or two that, if the time permits, I'll describe. So, what we work on is how leaves grow. Is probably there cannot be a simpler question than this one. And this is a photograph of a mature leaf. I mean, what question can you ask in a simple, unassuming, lackluster structure like leaf? It's not even a, leaf of an interesting novel like Harry Potter or something. It's a leaf of a plant. So I can actually, list 10 interesting questions that I can tell you that you can address on a leaf structure. First of all, how the size is determined. Why is it not bigger or smaller? How the shape is determined, why is it flat in structure, and so on and so forth. It's made up of cells. How do the cells know when to stop to grow, and why doesn't it continue to grow on, and so on and so forth. So first of all, this structure that you see is by and large very regular structure, almost like an ellipse. And when a structure is regular, that becomes beautiful. Why is it beautiful? Because it is it's a mathematical. You can predict there is a major axis, minor axis, and so forth. And when a structure is mathematical or regular, it inspires a lot of great minds to paint things. So what we want to know is, first of all, how this leaf grow. How did it come to this structure? What it is here? This is a mature leaf. It did not start its life like that. So if you take a photograph of a leaf at different time points, just like you are taking a photograph of a baby as the baby grows, this is what you will see. This is about 2 millimeter leaf, and this is the mature leaf. Okay, this takes about, for this species, about 15 to 16 days to grow. After that, the leaf will be there on the plant for another 3 months. Nothing, no other change will happen. The changing period is this one, from here to here, about 15 days. So how does this structure come to this structure? Okay? So the first thing what you see is that the shape is not really changing from the smaller to the bigger size. What is changing is the size. So if you take two leaves that I mark here and ask a question that how this leaf grew to this leaf, the question seems very stupid questions, because the shape does not change. What is changing is size. So one would say that this is growing uniformly or isotropically to a bigger structure, which may or may not be correct. How do you know? This is almost a homogeneous field. The field is increasing and then coming to this size. So what you can do is, you can do a very high-tech experiment. Since there is no external marker, you can put some external marker on the surface of the leaf, and then see that as the leaf grows, what is the fate of those marks. If the small leaf is growing uniformly to a big leaf, of course the dots will be spread on the leaf. This is a cartoon, of course, that is spread equally. And if this is the result, then you will say that this is growing uniformly. That is, each small region is growing at the same rate as the other region. However, there are other possibilities as well. This organ can grow only from the periphery, and the center may not grow at all. Then what you will see is that the dots are where they were, right? All the new structure has come on the periphery. So if you can do this simple experiment, you take a leaf which is still stuck to the plant, put some nice dots and all, and then it's a small leaf, and then let it grow. And as it grows and matures and becomes about 66 millimeter long, it won't grow anymore. What you see is the dots are not spread all throughout. The dots have come. So if I mark the dots here, which is here from top to the bottom, the dots are only up to this. So where did this region come from? This region came from this small region at the base, which is the growing region. So what does it tell you? This simple experiment. It tells you that the leaves do not grow uniformly. It has a gradient of growth, maximum growth here and minimum growth here. May not be linear or non-linear, most likely non-linear, but it's a non-linear growth. Maximum growth, minimum growth, or in other words, the cells are differentiating here and dividing here, because the organs grow by cell division. So if this region is a baby region, this region is a more mature region, then that should reflect in the cell that the leaf is made up of. So if you do a scanning electron micrograph and then look at the cells, you will see that these cells are dividing and very small. These cells are already differentiated. These are the stomata, that is the nose of the plant through which it breathes. And this is the more mature, and this is somewhere in between. So there is a gradient of not only growth, but there is a gradient of the cell division as well. And there are certain genes whose expression will tell you whether the cells are dividing or not. and his cyclin is one such gene. If you do, so this is a leaf which has been undergone lot of insults, experimental insults. Wherever you see some blue spots, that means the cells are dividing here. It's a marker. So you see lots of cell division here. There is medium cell division here and no cell division here. So all these three, what should I point probably here? All these three actually tells you that this leaf is not growing uniformly. It is growing in a anisotropic manner. This place is growing, this is less growing, and this is the least growth is at the tip. Now that one region of an organ grows differently from another region, or one part of an organism grows differently from another part, is historically has been called allometry. The botanists and zoologists have been working on this allometry for a long time, and a simple definition of allometry is the growth relationship of one organ to the rest of the body. And this is not so. Here this organ or this part is growing at a different rate from this part. So this is a classic case of allometry. And this is not true only for the leaves, it's true for us as well. We also grow allometrically. We are not a lot different from the leaves anyway. So once it grows and matures, you see that the line is here, because so this 50% is given to only this much and this 50% is given to this much is anisotropy. That's exactly how a baby grows. A baby, when it is, this is 8 weeks old, she is not born yet, the head is almost 50% or 45 to 50%, and the rest of the body is 50%. That's how we look actually at some point of time. We all looked at some point of time. So this is how the division, the head and the trunk is. However, when you grow to maturity, then our head is about 10 to 12%, 12 to 15%, and rest is body. The children interpret this result as the adults have smaller brain. Well, I have no problem with that, probably it is true, but what it actually means that our trunk or the body is growing at a faster rate than our head is growing, exactly what the leaves grow. And this is the classic case of allometry. You can actually measure this extent of allometry, and there is this great man, Julian Huxley, who actually measured it and quantified and then expressed what the allometry and how much allometric it is. What he did is, I am giving an example, that what he did is, he measured as a function of development or as a function of time, he measured one organ, and he also measured the remaining of the organs and plotted them as a function of the one to the other. The trunk in this axis, y-axis, and the head is x-axis, and obviously this is not a straight line because our body grows more than the head grows. So how do you write it in an equation? Of course, it's not a straight line, so you write it's a power law, y is proportional to x over alpha, where alpha is a non-unity, it is not 1. Of course, if alpha is 1, it becomes a straight line. And you can convert this into y equal to b, a constant into x to the power alpha. And I do not understand mathematics more than a linear equation, so I quickly convert into log plot and it becomes a straight line. So here it is y equal to mx plus c. So this is a straight line. What does this alpha tell? This alpha is interesting because alpha tells you that the extent of allometry, that means if our head grows twice, the body grows 2 to the power alpha, right? That's 2 to the power 2, that is 4 times, and so on and so forth. But alpha actually tells you much more than that, and I'll tell you what alpha tells. When alpha is greater than 1, it's called positive allometry. That means the growth is more in the basal side and the less in the top side. If alpha is less than 1, then it's called negative allometry. That is, our head is growing more than the basal side. And of course, if alpha is 1, it's a linear relationship, and there is no growth polarity, and then it's called isometry, a special case of allometry. So these 3 possibilities are there. Then why all of us and all the leaves grow grow only with positive allometry? I can't go around and measure the growth allometry of human being for obvious reasons. So what I thought that I'll ask this question in leaf and then see that why leaf grows from the base and not from all the 3 possibilities that are possible. So it's easy to measure the head and the trunk, right? Because there is a head and there is a trunk. But a leaf does not have a head and a trunk. But the good thing about leaf is they don't complain. You call the leaf that this is your head and this is your trunk. Okay, there's no problem at all. The artificial situation that you all do, and this is our x and this is y. And as we have seen that at maturity, this midpoint moves here. This is the growth of the x, final x, and this is y. Right? That we have seen. What you do is you put a dot in the growing leaf. First day x equal to y, and then keep measuring every day x1, y1, x2, y2, x3, y3, and so on and so forth. and then you just plot it into a log-log plot and see what is the tan theta of this plot, which will tell you the alpha value, or the if the top is growing twice, head is the y is growing 2 to the power how many times, or what is the alpha value. It's an easy assay to do it. So what we did is we went around and climbed a lot of trees in ISA campus, and then did about 80 to 90 species of different leaf shape and so on and so forth. We just completely ignored the leaf shape and other things. We took the proximal distal axis and then found out the alpha value. What we saw is, what was known is the leaves grow with positive allometry, and we found many of the leaves which grow with positive allometry, because the alpha value, which is this is the 1, and then all of these have the alpha value more than 1. As long as it is more than 1, we don't care how much it is. 4 is equal to 3, it doesn't matter, because as long as it is more than 1, is positive allometry. So it's just 4 means that is more allometric than 3, that's all. But however, we saw many other plants. So this is positive allometry, all right, which was known, which we also found that many of the species which produces leaf which grows with alpha value which is statistically 1, not more, not less, which essentially will tell you that this is actually grows with isometry and alpha value is 1. But there are some species where the alpha value is less than 1 and statistically it is significant, and this we call negative allometry. So there are species or the leaves whose leaf grows with diverse allometry, not only one specific growth polarity but different polarities. So let's put all of these species that we study and put together and see what the distribution is. About 50% is positive allometry, around 40% isometry, and then remaining is just ignore the mixed isometry, the allometry, and the remaining is negative allometry. So what we do is we pick the representative species from each group and then ask that how they behave at the cellular level. So we picked about couple of species here, couple of species here and here, and then see how they grow. This is a tobacco, which is a representative of the positive allometry that you have seen. This is a young leaf which is 38 millimeter long. The midpoint is here, and as it is mature, the midpoint moves up. It is a positive allometry. If you look at the alpha value is 4.4 here, and if you look at the cell size, since this is the growing region, you see the cells are very small and dividing and contributing to growth, and this is already mature, is not growing as much as the other end. So it is a positive allometry, and if you look at the cells, these cells, their mitotic index, which actually is a parameter which measures the rate of cell division. What I mean is the percentage of cell division. 5.5 means 5.5% of the cells are dividing here, 2.5% cells are dividing here, and 0% cells are dividing here. 0% cells are dividing here means it has stopped dividing and not growing as much. So this mitotic index also tell that this is the region which is growing and this is the region which is not growing. So basically this grows with positive allometry, and you take about a species, representative species from isometry, and then you do the study. This is a young leaf, you put the dots all along, this is a central point, this is x and this is y. At the end of the growth also, this is a bigger leaf which is x equal to y. So it does not grow from any polarity, it is a synchronous growth throughout the leaf. And look at the cell sizes, they behave the same way. That means the entire leaf is growing at the same rate and dividing. And if you measure the mitotic index, it is pretty similar throughout the proximal distal region, which means that there is no polarity of the growth here. So we have two different types of growths. And there is another one which is negative allometry. This is a croton, is probably there in all of your gardens, is a horticultural plant. In the day 1, which is 40 millimeter long young leaf, where the x equal to y, and you see at the end of the growth, the midpoint actually moves down. That means the top region has grown more than the bottom region, and alpha value is 0.6 as expected. Look at the cell size, these cells are dividing and these cells are maturing first, just the opposite of the positive allometry, and mitotic index also has changed, the polarity has changed. So if you put all these three together, you have three different ways to grow, the three different polarities of growing: positive allometry or growing from the base, negative allometry growing from the top, and there is no polarity at all and growing synchronously. And there must be some reason, there must be some molecular basis of that. So the hypothesis that your null hypothesis would be that there must be some sort of molecule, quote unquote, which will induce the differentiation, but at a different region. For example, for positive allometry, the red color is the differentiation inducing chemical, which will be distributed more at the top and less at the base. For the negative allometry, will be more at the base and less at the top, and here it will be equally distributed. So we did a screen of all the usual suspects, and none of them had any correlation, except there is one group of genes So this is a family of genes. Most of the important genes come in family, are not single. So this is GRF is a gene or gene family which promotes growth, and this is a top view of small tiny Arabidopsis plant, and this is a leaf, and you can see the distribution of this gene more here and progressively less here, and that's what you expect because this promotes growth, and Arabidopsis grows by positive allometry, that is more growth in the base and then less at the tip. So what we wanted to do is we wanted to see whether the distribution of this gene changes in different species, and then see whether that has any correlation or not. But there are many of them, so you can't go around some funny species and then isolate the genes from here. But luckily, all these genes are suppressed by one simple microRNA. MicroRNAs are a buzzword today. These are tiny RNAs which has a potential degrading factor for the genes. So this microRNA degrades all these genes and then do not let them function. This microRNA has an opposite expression pattern. This is again top view of a Arabidopsis plant, and this is the top tip of the leaf, and the base here you can see the distribution is more. And this microRNA is conserved, there is only single microRNA conserved in all the species. So what we thought is, we will cut the leaf of different species, young leaf, and then measure the microRNA here and here, and there are ways to measure microRNA in biology called northern blot. So see whether the distribution is changing in different species or not. So this is what is done here. This is a species which grows with positive allometry, that is immaturity here, maturity here, this is a growth gradient. What we did is we cut into two and call this is the tip of the leaf or head, and this is the base of the leaf or trunk, and then we see the distribution of the microRNA. This is the maturity inducing microRNA. You can see that there is a more microRNA at the tip which is more mature here, and less microRNA here which is immature. So here this correlation is there just like in Arabidopsis, but this was known, there is no problem. You can always tell me that you have the total RNA that you loaded here is more here and less here intentionally, that is why you are seeing that result, which is not true because there is another molecule which is equally distributed, so the amount of total RNA which is loaded is true. Not only microRNA is unequally distributed, that is more at the tip, the gene that is targeted by this microRNA which you isolated from this species, we see that we have the opposite distribution, more the microRNA. So you can see the target, and this is known to degrade. This one. So it has an inverse relationship. In this particular species which grows with negative allometry, that is maturity is here and immaturity is here. The expectation is there will be more microRNA here than here, which is opposite of this one. This is what you see, more at the base and less at the tip. And the target has opposite correlation, and here there is no polarity at all. So if you cut into two, tip and half is equal distribution, and the target also is equally distributed. So if you put all this together, what you see is there is a tiny molecule which is unequally distributed in different species and giving rise to different diversity in growth polarity. So I'll just leave you with one question, that if the leaf is growing really differently, for example, this species and these species don't really grow equally, right? They grow ununiformly, or they grow with a polarity. If one region grows more than the other region, then the leaf shape should change, isn't it? I'll explain this with this cartoon. So the question is how do leaves maintain flatness if one region grows more than the other region? It sounds weird because if this is a flat structure, and if it is growing uniformly, it will remain flat. There is no problem at all. However, if it grows ununiformly, for example, if the center continues to grow but periphery stops growing, it should bend to a structure which is like a cup shape, right? And this probably topologists call it a positive Gaussian curvature, and this is a zero Gaussian curvature because it is surface curvature. But by contrast, if the periphery continues to grow and center stops, it will become a saddle shape, right? And more importantly, especially during the tea time, your Pringle chips is a better example. So this is a negative Gaussian curvature, right? This is a surface curvature. So an expanding surface has the possibility of an option of remaining flat by growing uniformly, or being a positive curvature, negative curvature. This is the problem that leaf also faces when it grows from the small to big in the planar axis. How does it maintain the flatness? Most of the leaves are flatness. However, if you mutate one or two genes, which are you have to know which those genes are, it actually can make the curved structure. This is a sort of by and large positive curvature, not quite Gaussian, and here this is a negative curvature because the periphery is growing and it is going in the z-axis. That's why you see it is actually not a serration, but it is going it's a wavy. So this has a negative Gaussian curvature. So there are factors which are making sure So the gene, one particular gene is not functional here. Out of 26,000 genes in Arabidopsis, all are functional here. One of them is not functional here. Another one is not functional here. So you can remove different genes and you can make different structures which has some sort of a different mathematical. How do I actually measure that this has positive Gaussian curvature, this is negative Gaussian curvature? I'm done. So this is how do I measure? The best is that quantify this by laser scanning or something which we could not. But there is an empirical way of measuring. You use the Euclidean geometry. So in Euclidean geometry, we have studied that for a circle, if it is flat and if the r is the radius, then perimeter is this and area of this, that you have studied in the 6th standard or 7th standard. But if and only if this is flat. If this is not flat, this actually doesn't come true. So what you can do is you can make some modification with this one and you divide the perimeter by a square root a, this, which will get rid of r, and then you get a constant value 3.6, right? So now the perimeter, the p by square root a parameter, which I call curvature parameter, will continue to remain 3.6 no matter how big the circle is, as long as it is flat, right? So what I can measure is, I can measure the flatness of this, the p by square root a of these leaves, and then see whether it is 3.6 or not. So the possibilities are, if it is flat is 3.6, if it is positive Gaussian curvature is less than 3.6 because the perimeter is less and then the center is more, and here it is the negative Gaussian curvature is more than 3.6. You do exactly those measurements. This is the wild type. It's not exactly a complete circle, but when you measure the perimeter and p by square root a, it is 3.8, little more than. However, this is a circular leaf. Still the p by square root a is 3.2, which is significantly less than 3.6, and if you measure the p by square root a here, it is 4.4. So there is decrease in Gaussian curvature, there is increase in Gaussian curvature, decrease in p by square root a, increase of p by square root a, which is a sort of empirical expression of the So we sort of identified few of the genes, and the more description I will not tell you here because he has already stood up. I will come to the end of the talk, and then well, I would rather ask these guys to thank me because their work on their behalf I am presenting here. And one more thing, so this is I would dedicate this one to all the flat structures on the earth, the biological flat structures. This is also, it can be compared with the leaf, but the basis of the flatness of Drosophila, which is probably the best model system on earth, genetic model system, has not been determined, but at least the plant science has beaten the animal science in one aspect, that is in determining the flatness of the flat structures. Thank you.